Good morning, everyone. Here we are at week number 6. So let's take a look at what we did last week and what we're going to do this week. Last week we learned about DNA, deoxyribonucleic acid, and the importance of it, the fact that it contains all the traits, the information to make all the traits that make us organisms. You also learned about PCR, which is the polymerase chain reaction. And that's a technique where DNA is copied over and over and over again, millions or billions of times over until you get enough copies of target DNA to actually do something with it, test it, look at it, etc. So what are we learning about this week? This week is all about genetics. So genetics is how traits are inherited. And of course, genetics is connected to DNA because the genes in DNA are where all the traits are located. So genetics is how you inherit these traits. And you've heard of some of this before, and you know some of this. When you look at your biological parents, you can see traits in you from each of them. Also, you know some of these terms already: dominant traits, recessive traits, sex-linked traits, etc. So I know that you know some of this already. The scientist that really began working on genetics was Gregor Mendel, who was an Austrian monk in the 1800s, and he studied all about pea plants. So you might ask yourself, well, why in the world did he study pea plants? Because farmers grew peas, and there were lots of varieties of these peas. And Gregor Mendel had a little garden plot, and he planted pea plant after pea plant after pea plant, and studied the genetics. And he came up with the terms dominant traits and recessive traits because that's what he could see. Sometimes traits were hidden in a generation and boom, they would pop up in the next generation. So he studied a lot about genetics and gave us a lot of what we know, but of course we know more now than we did then. We know what DNA is and what genes are and how traits are found in the DNA. We know all of that now. An interesting fact about Gregor Mendel, he studied thousands and thousands of pea plants and collected a lot of data on these characteristics in peas. And as soon as they bumped him up to an administrator, all experimentation stopped, all of his science stopped, which is a shame. So he quit doing his science and he was an administrator at the monastery. So don't let that be your life too. I mean, really, that's sad. Stop doing what you love to do and become an administrator. Yeah, you're going to make a lot more money, but are you going to love doing it? Anyway, just my commentary for today. You'll be doing some Punnett squares. So Punnett squares are an organized way where you can put traits from gametes are sex cells in parents, and put those traits from the gametes into possible offspring. And I will have some examples of that. I will work some out for you in some videos that I will post. We're going to do monohybrid crosses. And so this is where each parent has one of each type of trait. One dominant trait, one recessive trait. We represent these with letters. So you can see in my example here, capital T, little t. Capital T represents the dominant trait, T, whatever it is, and lowercase or little t represents the recessive trait. And you're going to take two parents who are hybrids. That means they contain the two different traits, and we're going to cross them and see what we get. In the experiment that you do this week with the plants that you're growing, this is going to be a dihybrid cross. We're looking at two separate, distinct traits, and we will see how they are inherited in the offspring. All right, that brings us to the experiment that you do this week, the germinating seeds lab, which is you're working with fast plants. They're called fast plants because they grow very fast and you can look at their traits within a matter of days. But we are growing two complete sets of these plants. So you have to start today, if not yesterday. So you have to start right away or you won't get finished with this. Okay, so start your germinating seeds right now. This is not difficult. Most days all you do is check to make sure that there's water on the filter papers like you're supposed to have to keep your seeds and plants moist, but you have to get started now. I have 3 Punnett Square videos. They go 1, 2, 3. They're back to back that show you exactly what is happening in the genetics of this lab that you're doing. Please, please watch the Punnett Square videos. I will put them in the announcements in a separate announcement today. But they will help you with your lab for the germinating seeds. I work out the Punnett Squares. I talk about the traits. I give you hints about things that you may not realize when you read that lab. There's certain portions of that that are extremely important. For example, the 2 parents that were used to make the seeds, the first seeds that you will plant, not the parental one, but the F1 generation, those parents are called true breeding plants, and that means that they have 2 copies of the same gene. So they don't have big T, little t. They have big T, little t, little t. That's what true breeding means, and you may miss that. So watch my Punnett Square videos, please. The 2 traits that we're looking for are height, which is why you need to grow these seeds for 4 days. If you don't, if you grow them for 2 days, there's no way that you can tell the difference between a dwarf plant and a normal height plant. 4 days you have to grow them, okay? Height and also stem color. So we're looking to see whether the stems are purple or non-purple. Non-purple is anything other than purple, which is going to mean green. But here's a hint for you. If you see purple anywhere on that plant, if you see purple leaves, partial purple on the stem, if you see purple at all, that is a purple plant. And that is extremely important because some students think, well, the whole stem wasn't purple, so it's non-purple. Nope. If you see purple anywhere, it is purple. Okay, and the second thing that you have to do is a worksheet. It's the biology project worksheet with some problems on it that you have to solve. Most of the time, well, some of the questions require you to write out Punnett Squares, another reason to watch my videos. So you have to answer these questions using Punnett Squares and what you know about genetics. Now, the instructions say to go to the biology project and work all the problems that they have there. That's a lot of problems. That's a lot of work. If it were me, I would open this worksheet, read a problem, go back to the tutorials and the example problems at the biology project, and look up what I need to look up right then and figure out how to answer each question. Otherwise, it's going to take you a very long time. But you do it the way that you want to. That's just a suggestion. You may write out the answers on this worksheet, like write by hand. But if you do, make sure that I know the difference between a capital T and a little t. Uppercase letters, lowercase letters. If I can't tell the difference, then it's wrong. Okay? I can't guess what you know. You're trying to show me what you know. Okay? When you talk about gametes, when you write gametes, when you type gametes, you need to draw a circle around your gametes. You can do that even with a Word document. Try your best to do that because that shows me that you know what the gametes are. Remember, gametes are those sex cells. Okay? You can also take a look at my Punnett Square videos because I will show you exactly how to do this. And another thing, make sure you space out your answers. So this worksheet has all the questions all jammed up one right next to the other. Put some spaces in there. Give yourself enough room to do your work and to type out or write out your answers. Make sure that your answers aren't too crowded. That's for you and for me. Okay? And as always, if you have any questions, just let me know. m.sigmon@snhu.edu. I'm here for you. You probably will have questions this week. And I'll also have office hours next Sunday evening, 6 to 7 Eastern Time, in case you have questions and you can't get something answered on these, either the worksheet or your experiment. Okay? I hope you have a great week. There's lots to do this week. So I hope you have a great, great week. These are monarch butterflies that are migrating. I just saw them the other day, so I put the picture here. Anyway, see you soon.